Okay, so we are live, just between us.
Can I ask everybody to like, if they have questions, to just unmute themselves and ask the question as I'm speaking, so that we can have more of like a conversation. Is that fair, Sally?
That's perfect. Yes, it's a small group,
awesome.
so that will be awesome.
Yes. Okay, so
I'll let you
yes.
already speak.
No, go for it.
Clarice, thanks a lot for being here. So the floor is yours.
Thank you. No, it's been great, and Araceli is one of my favorite quantum people. So it's thank you. It's like, I hope we keep on talking and coming up with ideas of what to do. So thanks for the invitation. So I like to call myself a quantum engineer. So this means that I build apparatuses to study and control things that are so small and so well protected from their environment that they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that ruled everything big around us. So people get surprised when I say that I study things that happen in birds or butterflies. So what do I want? So before I explain where I'm going, I really need to tell you where I come from. And we're going to start by talking a little bit about hardcore quantum mechanics and quantum sensing. And by the end of the talk, we have talked about things of biological relevance, such as organismal migration, how our bodies respond to oxidative stress, radiation. And I hope I will have motivated a little bit how I think that we can learn with nature to build better technologies. And that's because I'm a quantum engineer who's interested in how quantum physics informs biology at the nanoscale. So let me start then this weird talk by saying that I think that humankind is obsessed with measuring things better, because it means, or it maybe means, that we can understand nature better. So we can think about measuring better frequencies, for example, with this atomic clock that measures very well what a second is based on atomic transitions. We can think about measuring better magnetic fields so that the image of our baby is better resolved. And yes, that's a magnetic resonance image of our baby inside the mother's belly. And we can also maybe think about measuring better accelerations so that our gaming experience is enhanced. Those tiny little accelerometers are now ubiquitous in all our handheld devices. But the question that I ask is, what happens if the quantity that you want to measure is very, very small? Or worse, what happens if the object causing the quantity to be measured is very, very small? I'm going to argue that in this case, you need a tiny little sensor that can measure tiny little things. If the sensor is tiny, let's make it very tiny. Let's make it quantum. And there's actually a whole body of mathematical evidence that can prove that if you use a quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. So I'm going to talk about a technological quantum sensor that I've worked with in the past. In the past, I put a single electronic spin in diamond to work as a quantum enhanced magnetic field sensor. So that's what I promised, but before anything, I would just like to remind you of what a spin is, right? So a spin is a merely quantum mechanical property that has no classical equivalent. Electrons have spin, some atomic nuclei have spin, and spin measures how well this quantum object interacts with magnetic fields. Physicists and engineers and chemists, they usually depict spin with an arrow up or arrow down. Spin up or down usually represent two different energy levels of this merely quantum property of spin. The single spin in diamond that I put to use as a magnetometer arose in a crystalline defect in the diamond lattice. Okay, this crystalline defect is actually a color center. Color centers are crystalline defects that absorb light and then fluoresces and then emits light. And in fact, color centers are responsible for the color of those beautiful diamonds that you see there in the picture. The particular color center that I worked with is called a nitrogen vacancy center. It arises naturally, but it can also be engineered. And it happens when one vacancy, one missing carbon atom, sits next to a nitrogen, which is the most commonly occurring substitutional impurity in the carbon lattice. When those two things come together, the nitrogen impurity and the vacancy, there's like, it's like a mess of electrons interacting. Near the vacancy, there are plenty of unpaired electrons. There might be extra charges. The nitrogen has an extra electron, right? But in this mess of electrons, something very nice happens as you calculate the quantum mechanical energy levels of this compound. And almost magically, the picture simplifies a lot, and the energy levels of this electronic mass looks like the energy levels of a single electronic spin 1. Okay. So effectively, you can look at this atomically sized effect and think of it as a single electron spin. And the way we play with this spin and find those nitrogen vacancy centers in diamond is as follows. You take your diamond sample, your crappy diamond sample, actually the very first diamond chip that we worked with was a discard from a jeweler. Okay, so at room temperature, you put your diamond chip on top of a moving stage, and you start moving this diamond around at the same time that you shine a laser of a particular frequency onto this diamond slab. So we actually do this with lasers because it's more efficient and more convenient in the lab, but it doesn't need to be a laser. But we shine light of an appropriate frequency that can be absorbed by those color centers. So in the minutes that as the slab is being scanned and the laser hits one of those centers, we expect, because it's a color center, we expect the center to absorb the light and then fluoresce back, emitting light. And this emitted light we can actually collect. So this is what the emitted light looks like at increasing zooms. Okay, to the bottom right there, what you see is a single blob. This blob is about 500 nanometers in diameter. It's a big blob, but again, it's super impressive because this is the fluorescence signature of an effective single spin in the diamond lattice. Again, the blob is big because it's diffraction limited, but the fact itself is atomically small, right? And yet you can capture its fluorescence signature in the computer with a very simple microscope setup. But there's something even more special about this defect, which is the fact that using quantum jargon, it has a quantum state dependence fluorescence intensity. This means that just by looking at how strongly this blob is emitting light, you can actually tell if the spin was up or if the spin was down. The mean emitted fluorescence level of the spin in state up is different from the mean emitted fluorescence of the spin in the state down. So just looking at this very convenient experimental parameter, the fluorescence intensity, we have quantum information on the state of the spin.
Can I ask you something?
Awesome, always.
Yes, this fluorescence intensity doesn't depend how deep is this NV center in the diamond?
It does.
Okay.
It does, right? But once you have singled out a center that does not move, then you can actually calibrate what's the mean intensity for the spin up and what's the mean intensity for the spin down.
Okay.
So
and when you're saying it doesn't move, does it mean that when you shine the light, actually you can excite this electron and it doesn't move in the frequency of the movements of the crystal?
So this defect is, if you will, nature's own trapdion. So the defect is not going anywhere at room temperature. At room temperature, this compound is stable. It doesn't move. It doesn't go anywhere. The single electron is there. So it's there for us to play with and to do things for us. And that's very convenient too, right? But you're absolutely right that every defect fluoresces at a different mean intensity, but once you fix your attention onto one, you can actually calibrate what's the intensity for spin up or spin down.
Thank you.
But I did promise you that we were going to make a sensitive magnetometer out of it, and here's how it works. So here you see here I depict just the two different spin energy levels, up or down, that here I call 0 and 1. And it turns out that the difference in energy between those two levels is in the microwave. It's about 3 gigahertz. Okay. And it turns out that one of those states, the state that I marked with 0 there, is not sensitive to magnetic fields, in that if this defect is immersed in a magnetic field, state 0 does not care at all. Whereas state 1, on the other hand, it is sensitive to magnetic field, so that if it's immersed in a magnetic field, the state 1 gets a jump in its energy by a tiny little amount that I'm calling delta. And it turns out, for those of you who might remember, this delta amount, this detuning delta, is proportional to the magnetic field intensity via the Zeeman interaction. So here's a point that I'm going to make that is very important, that doesn't often get made, which is the following. The problem of measuring magnetic fields is then mapped onto the problem of measuring tiny detunings delta from a known resonance, okay, from a known energy difference. And in fact, this is how mostly quantum sensing and quantum computing works. Many times what you're measuring is the tunings, like slightly little deltas from a known energy difference. And very conveniently, measuring the tunings from a resonance is something that engineers and physicists have known how to measure for many decades. Okay, so many quantities can be measured in this way. It's not only magnetic field, it can be electric fields or temperature that cause different shifts from a known resonance, and people know how to measure those detunings. So we can measure tiny magnetic fields with this nitrogen vacancy center because experimentalists know how to measure detunings from resonance. But there's a catch to this. The catch is that this way of measuring quantities, of measuring magnetic fields in this case, only lasts during the time for which this spin is well described by the laws of quantum mechanics. And maybe you remember from your quantum classes that everything that starts quantum dies classical, right? This is the reason why quantum computers cannot be operated at room temperature. Usually they have to be operated in vacuum, under very low temperature conditions. Everything that starts quantum at some time will interact with its environment so that it stops being quantum. For the case of this spin, for a short time, it can be well described. This spin state, if you remember from your quantum classes, can be described by a coherent superpositions of states up and down, which is not trivial and not classical. But after a short time, this spin will have lost all its coherent superposition superpowers, and it's going to behave as a classical tiny needle that is either up or down. This time during which the spin is quantum is usually referred to as the coherence times. Okay, and what's very special about this defect in diamond is that at room temperature, in the solid state lattice, right, this spin has a coherence time of about 2 microseconds, which is unusually long. Okay, so the whole the name of the game in quantum sensing is like, can we keep this spin alive as a quantum object for longer and longer, so that we can play with it as a magnetometer for longer and longer. You can think about it as a signal processing problem, right? If you have a signal that you can acquire as a function of time, the longer you can acquire the signal, when you Fourier transform it, the better your detail in frequency is going to be. So here it's sort of the same thing. When the spin is no longer well-defined quantum mechanically, we cannot acquire this quantum signal anymore. So what we want is to keep this spin as a quantum object, and people know how to keep spin alive for longer, how to counter the effects of decohering noise by applying engineered electromagnetic fields. And people have known how to do this since the starts of, say, magnetic resonance experiments in the middle of the past century. And applying engineered electromagnetic fields is akin to giving kicks onto someone on a swing, right? So that this swing keeps on, like, going back and forth in a, so that the swinging amplitude does not get damped. So this is, so there are particular ways of how to apply engineered electromagnetic fields so that your spin is kept alive for a little bit longer. So I'm going to show you one example on how to give those kicks to the spins. It's going to be probably the most technical part of my talk. So before anything, I just want you to keep in mind the big picture, okay? The big picture is we want to have a tiny little sensor that we can bring in proximity to a tiny little sample and be able to measure tiny little magnetic fields produced by the sample, because we know how the sensor itself reacts via detunings to the magnetic fields of the sample. So this is the big picture, okay? Don't forget the big picture while I talk about the technical details, but I want to show you one example of such a spin control experiment. So in my past life as a PhD student, we gave kicks onto this spin so that it worked for a longer time as a quantum magnetometer using what's known as a rotary echo sequence. This rotary echo sequence is depicted in this plot here in nuclear magnetic resonance language. So let me translate it to you. This means that I'm applying a microwave resonant with the 0 to 1 transition in the presence of any external field onto my spin. I have a cable that's providing microwaves onto the spin. And the microwave is a CW, it's a continuous wave, but at particular intervals that the experimenter can choose, I am flipping the phase of this microwave field, okay, by 180 degrees. I'm doing a phase flip. So there's always microwave coming in and I'm flipping this phase. This pulse sequence, this particular engineered electromagnetic radiation has been known for 3/4 of a century now. And this sequence is known to correct for imperfections in your driving fields, in this case, for imperfections in your microwave. But the sequence is also known not to correct detunings from your resonance. And if you remember my previous slides, I just told you that by measuring the detunings from the resonance, we can get magnetic field information. So that's good. Let's see what this. Let's see what signal I get from my spin once I apply this electromagnetic engineered excitation onto my spin. I get something that looks like this. The signal is just like a fluorescence intensity normalized. So when the fluorescence is at its maximum, the spin is definitely in the state up. When the fluorescence is at its minimum, the spin is at spin down, and anything in between means that the spin is in a coherent superposition of up and down. We can apply a formalism known as average Hamiltonian theory to understand how the signal actually depends on the tiny detuning that we want to measure, this detuning delta here. So if we Fourier transform this signal, we get something that looks like a spectrum. I'm not explaining to you how to read the spectrum, but you have to believe me that with this signal that you see there, we are able to read via this detuning delta magnetic fields that are as tiny as 1/10 of the magnetic field of the Earth. Okay? So this is just one way on how we measure those detunings in the lab. But again, don't get bogged by the math here. Don't forget about the big picture, which is having this tiny sensor that you can bring very close to tiny samples producing tiny fields. And now I'm going to make a pause for questions, but before I make a pause and ask for your questions, I just want to make you note that I've just shown you a quantum sensor that works at room temperature and in noisy environments. And in particular, the transition energies between spin up and spin down, I mentioned that it was about 3 gigahertz. If you just Google very quickly what thermal energies are in kBT, right, are in gigahertz, about I think 6,000 or so, right? So yes, what I'm saying is that there is this quantum sensor that has a tiny energy compared to the thermal background that still can yield important sensor information during the time while it's well described by the laws of quantum mechanics. And this despite the fact that its energy is smaller than thermal energies. All that's important is the time before thermalization, which is basically this coherence time, which in the case of this nitrogen vacancy center for a crappy diamond is about 2 microseconds. So what are your questions?
Can I ask again? Sorry.
Always.
Yeah. I have kind of 2 questions. So do you need, obviously, when you do this experiment, to isolate this from the environment, I guess, because if you, the magnetic field on the Earth, you said is 10 times more than this, what you can detect, right? So do you need to do some special setup to isolate this from the
so it depends
environment?
It depends on what you want to measure, right? If you know that your sensor is fixed, you can measure the magnetic field of the Earth and then like subtract from whatever it is that the spin is seeing, right? If you have it in a deployable instrument, you're right that you should first of all measure the magnetic field of the Earth and correct for that.
Okay.
But in some
so I was thinking more or less, if you try
yes.
to put this in quantum computer and you want to measure the bits, so basically what you're trying to measure is qubit, right? So if you try to bring this in the downs in the minus on 4 Kelvin and so on, and you measure qubit, let's say spin qubit.
So I'm
can you do this?
that we actually build a quantum computer out of this at room temperature. We can make a quantum computer that works with spins at room temperature. I mean, this is okay. There might be reasons why this might not be ideal. So quantum computers working at low temperatures and in vacuum perform much better than this currently, but this is a route for room temperature quantum computing. And moreover, you can do, and I think that's one of the most interesting things about this nitrogen vacancy center, you can do sensing experiments at room temperature. You can actually put those things inside biological specimens. You can take those things to the field and you don't need cryogenics or anything. So,
okay.
So I think that this is a very good quantum sensor architecture in general.
If I may,
Okay, so which brings me to the...
Sorry,
yeah, sorry.
just to tell you, there are 2 or 3 companies around the world who are actually trying to build quantum computers based on NV centers and other kind of a room temperature platforms. So
okay,
yeah.
so yeah, that's fine. So from the point of view, this was related to my next question. So you have a crystal which is how big is it as a dimension?
It doesn't matter. So we had a tiny little chip, but people now can fabricate nanodiamonds from 5 to 15 nanometers in size that contain
Yes.
exactly one
Okay.
of those centers.
Yeah, so this was my question. When you measure this and you say it's one spin, how do you know it's one spin? I mean, how you're sure that you have one IV center?
Yes,
NV
I
center,
didn't
not
tell
5 or
you
50?
because we can do photon correlation experiments and show that there's a single emitter there. So the moment when I
Okay.
get to focusing on one blob, right, I can
Okay.
do single photon correlations that prove that there's only one.
Okay.
No two
Okay.
photons arrive at the same time.
Okay.
But you do have to test for that.
Okay. Okay.
And the other thing I wanted to mention is that, and Araceli, of course, knows way more than I do, but there's also some companies that are trying to make magnetic sensors, magnetic resonance healthcare sensors that are also based on those NV centers. There's a company that is doing some sort of diamond dust. They spray something with a lot of those centers, and based on their spin signatures, it can give you like a very hardly falsifiable information on it were a barcode or something. So there's very interesting things that people are doing commercially with those nitrogen vacancy centers now.
Okay.
More questions.
Sorry.
No, sorry. That's what keeps me going. More questions.
You said about the sensitivity that you measure a tenth of the Earth's magnetic field. Did I hear that correctly?
Yes, 1/10.
Like 5 millitesla or something like that.
Yes. No, 5 microtesla.
5 microtesla, yeah. So that is, of course, it's fantastic because it's at room temperature, but it's not as good as atomic chip scale. What
sure.
Could you get better?
So for sure, this was done in a very crappy sample, right? That wasn't prepared in any way. Something that people are doing is they're preparing isotopically pure diamonds. That is diamonds with fewer carbon 13s, because carbon 13s have spins that contribute to the spin noise, contribute to the coherence. So people are doing isotopically pure diamond. People are perfectioning decoupling sequences or those engineered pulses to keep your spin going. So this by no means is state-of-the-art sensitivity of what people can get with those nitrogen vacancy centers, but even with a very crappy diamond, which is naturally occurring, you already can sense very tiny magnetic fields. But there's no doubt that other magnetometers are better, and everything has their trade-offs, right?
Absolutely.
And people especially like this because you also can insert it in bio things, right? Which is very convenient.
That definitely don't want to do with your atomic chip.
Yes.
Also, people often talk about sensitivity in terms of per square root of hertz. What is your scale for what you're doing?
I don't remember. If someone
that doesn't Google. Don't worry. I'm just wondering because you probably need some length of the signal in order to do your Fourier analysis.
So we never acquired for much longer than 5 microseconds.
Okay.
I am sure people now are doing nanotesla per square root hertz. I don't think it's what we had. I don't recall the exact number that we got anymore, but that's absolutely right. Usually sensitivity should be given in Tesla per square root hertz, because this, for those of you who have never heard of it, also reflects the time that it takes for you to take your measurement, which means that you can have the best, the best sensor ever, but if it takes you an infinite amount of time to get that information, it's not a good sensor after all. So this sensitivity per square root hertz weighs a little bit like how much time you actually need to perform the experiment. So yes.
Thank you.
But like all of this was actually a preamble to talk about things that I really want to talk about and maybe pique your curiosity a bit. So up to like a couple of years back in my life, I was used to dealing with sensors like those that you see there, right? Like humankind made sensors. And yes, I plot there a big magnetic sensor, like conventional magnetic sensors. But at some point, and more or less by chance in my career, I sort of realized that nature many times makes sensors that surpasses humankind made sensors in crazy ways. And I would like to talk to you about a naturally occurring magnetic sensor that we have reason to believe is quantum driven. And we'd like to again talk to you a little bit about that and where we are going in this space. And this is what I'm really excited in these past few years. So, but in order to start talking about biological sensors, I'll have to talk a bit about biology at the nanoscale, namely the chemistry. So it's been known for many decades now that magnetic fields can alter the final products of a class of chemical reactions that are spin sensitive. This means that there are chemical reactions that depend on spin in the following way. At some point, there is a loose spin that can interact with magnetic fields. And if this spin ends up to be up, the reaction continues through one pathway. And if this spin happens to be down, the chemical reaction continues through another pathway. Importantly, the macroscopic final products of this chemical reaction can be different. And even if the spin phenomenon is something that happens very fast and at the top of a chemical reaction, its effects can be again sensed macroscopically and at much longer time scales. Okay, and the point that I hope you see that I'm getting at very soon is that, well, if we can somehow control this spin at the stop of this chemical reaction in the same way that we can control spins in diamond, we might be able to affect the fate of a chemical reaction. So this fact that magnetic fields can alter chemical reactions has been demonstrated at room temperature, in solution, in the gas phase, in the solid phase, and down to magnetic field strengths of the magnetic field of the Earth, which, as we said, is about 50 microtesla, which is actually, I think, orders of magnitude tinier than the magnetic field that you of the magnets that you put on your fridge, and also tinier than the magnetic field that you sense when you hold your cell phone close to your face. Okay. Here's how this notion started appearing in biological contexts. In the beginning of the 80s, people wanted to understand how birds migrate. And it's definitely known that birds do use the magnetic field of the Earth to migrate north, south, even if it is only as a partial cue. There's no doubt about that. And people really didn't know how that worked. People tried placing magnets onto birds to see if they got disoriented, and none of the hypothesis was working. And then a theoretical biophysicist came with a hypothesis, okay? And the hypothesis was that were the same type of spin-dependent chemical reactioning happening in vivo under physiological conditions in migrating organisms, organisms could sense magnetic fields to the extent that it could sense different physiological concentrations of products coming from these spin-dependent chemical reactions. Okay? And actually, the hypothesis was that this is not proven at all. This is just a conjecture. People thought that for birds in particular, maybe there was some sort of a modulation of light sensitivity as a function of magnetic field that happened in the retina of birds, so that it's still hypothesized that birds might see magnetic field lines as they move their heads around, so that they could maybe even see like signatures of magnetic fields in their vision. Okay, so it was at that point an outrageous sort of idea, but people did start looking at things that proteins that could sustain this sort of spin-dependent chemical reaction. And it turns out that at that point, the only animal protein known to be able to sustain these spin-sensitive chemical reactions was a photoreceptor called cryptochrome. Cryptochrome is it's a fluorescent protein, is a photoreceptor. It absorbs light and it fluoresces, and it's present in the retina of birds, in the antennas of migrating butterflies, in our eyes. And it's actually, and I think biologists have never corrected me on this, and I think this is true. Cryptochrome is a fluorescent protein that is present in all our cells because it has circadian rhythm regulation functions. And as people started researching more about cryptochrome, people saw that cryptochrome was present throughout the tree of life, and that many organisms seem to be displaying some magnetosensing behavior. The evidence was not complete, but there seemed to be some effect on magnetosensing that involved cryptochrome. What are your questions now? I see that you have your camera on? No? So again, it has started to become established that magnetosensing via cryptochrome was maybe a thing. And yes, right there, there have been some studies involving humans and cryptochrome as well. The evidence for this cryptochrome-based magnetosensing is really, really out there, but unfortunately at very disconnected length scales. And this is a problem. So on the very tiny length scales, as you see to the left of my slide there, in the chemical length scales, it has actually been shown unambiguously that cryptochrome works as a quantum sensor in the very same way that the spin in diamond works as a quantum sensor. Okay, for example, this is the top left plot there. Researchers tracked the fluorescence emitted by cryptochrome as a function of the time during which they excited cryptochrome of an appropriate, excited the cryptochrome with an appropriate laser. Okay, so cryptochrome, just like a color center in diamond, is a fluorescent protein. It absorbs light and then it fluoresces. So what you see there when you excite this protein in a vial with laser, the first thing that you see is that the fluorescence goes down. You're killing the pigment. It's called bleaching. But very interestingly, as the researchers post a tiny magnetic field on and off, and this is not an artifact, it has been proven using different magnetic fields, the fluorescence started being modulated up and down. In the same way that just by looking at how strongly the nitrogen vacancy center was fluorescing, you could say if the spin was up or down. Just by looking at how strongly this cryptochrome was emitting light as a function of the magnetic field, you could actually distinguish like if the chemical reaction took place via the spin up pathway or the spin down pathway. Moreover, and this is not super established yet, but there's good evidence that coherence times for the electron in cryptochrome can approach 1 microsecond at room temperature. Okay, and again, I quote the 2 microseconds that I quoted for a crappy naturally occurring diamond. So it's not that far from that. And again, there are better ways of estimating this, but you can sort of do a back of the envelope calculation. So how long would you need something to be coherent to feel, for example, the magnetic field of the Earth, right? So here's the back of the envelope crappy estimate, which is too long, but anyways. Electrons in the presence of a magnetic field, electron spins, they're going to precess around the field. And the precession frequency is also known as the Larmor frequency. One estimate for if you can sense the magnetic field, one magnetic field, it means that you can precess, say, once around its Larmor frequency to feel it, right? So the magnetic field of the Earth is about 50 microtesla. For a free electron, this gives a Larmor frequency of about 1 megahertz. And the time that it takes for one full precession at 1 megahertz, 1.4 megahertz is about 750 nanoseconds. And again, this is a little bit shorter, but on the same order of magnitude that I mentioned, that cryptochrome might be coherent for up to 1 microsecond. So this is super cool, right? This in solution is behaving as a spin quantum sensor, a biological quantum sensor. However, the next step in the length scales where people make those experiments to see if those things can happen in vivo, the length scales are huge, right? People do experiments with whole birds, with plates of thousands of cells, with flies. And even though the results of those experiments are consistent with the theory, the spin theory that is confirmed by chemistry, it's very hard to say, well, the bird is doing this because it's quantum for a short time, right? There's some quantum process effectively doing something inside its eye. So let me go through some of the evidence that is there, but I'm going to argue that it is not enough. So people put birds inside big cages with magnetic shields, and then they apply magnetic fields inside. And during migration season, by changing those magnetic fields, they can change the direction that the bird wants to fly out of the cage. Researchers can train flies. I have no idea how that's done. This is like magic to me. Researchers can train flies to find food based on the presence of a magnetic field. And then the flies are trained to find the food given the magnetic field. And then the researchers remove the cryptochrome gene from the flies, and the flies no longer can find the food. And then in a further experiments, the researchers put inside the fly again human cryptochrome, and the flies are back to being able to find the food, right? So all of this is consistent with what comes from the spin model, but people don't talk to each other, right? Like people are not convinced either way, and I get it, because there's such a disconnect between those length scales. So here's what we're starting to do in our lab. Our idea is to sort of bridge those two length scales and start doing quantum sensing, quantum enhanced measurements in those spins and cryptochromes inside, say, single cells. So to cut a long story short, we're building glorified microscopes with coils, okay? And what we want then is to use the machinery developed to study and control technological quantum sensors and start applying them directly onto those living quantum sensors. In other words, treating them as bona fide quantum sensors, because we're convinced that this is what they are, right? So we want to start to apply engineered electromagnetic pulses so that we can drive those spin around and study them, right? And again, we think that this field of quantum biology, I think that its bottleneck is really that it's like high-tech experiments that are quantum inspired and that try to treat biological entities as quantumly as possible. And then you might say, well, but why do I care, right? It's only birds migrating, and actually it's not. Again, there's evidence at those disconnected length scales, but evidence that the same sort of spin-dependent chemical reactions might be altering important things for cellular metabolism, how much cells do respiration, how cells regulate their pH, cell growth regulation in planaria, the yield of DNA repair. And I don't have it here, but a friend of mine in Munich, he's a precision measurement physicist. So for a living, he builds hypomagnetic chambers, which are chambers that shield the magnetic field of the Earth down to like nanotesla. He does that for a living, and then he puts precision like ultra cold atoms experiments inside to measure very, very fine things. So because he was starting to hear things about magnetic fields, what he and actually a group in Mainz did in 2 different of those hypomagnetic field chambers was the following. So it's 2 chambers doing this experiment under the 2 conditions that I'm going to describe. They decided to grow tadpoles inside these chambers for 2 days, which it was the longest that they could do without asking for a license to do bio stuff, under 2 conditions. One only with this nanotesla residual field, and another one with like an internal magnetic field that mimicked the magnetic field of the Earth. Whereas the tadpoles inside the mimicked magnetic field of the Earth were okay, the tadpoles under this nanotesla field macroscopically were about 30% malformed. Okay, like macroscopic, nothing genetic. You looked at them and you could see that they were like all weird looking, not fully developed. And this is not evidence, an ambiguous evidence that this is spin mediated, but given the fact that tiny magnetic fields are making such a huge difference in development, and given the fact that it's not even like adding the magnetic field, it's like taking away the magnetic field, right? So my best guess is that this is a spin mediated process, and it brings all sorts of questions, right? Not only for biologists, but also for like people who are concerned with space travel, right? Like, what's the magnetic field on Mars? Can we reproduce? Can we grow lettuce on Mars, right? Or when we have astronauts doing multi-generational travel in space, what sort of magnetic fields are they exposed to? How does that affect cellular functioning? So yeah, and what our group is trying to do is, again, in a very high-tech way, either establish or refute the extent to which quantum physics might be playing a role in physiology, and to which extent it can be manipulated, like quantum physics and biology being manipulated to technological and therapeutic advantage. For example, if nature is really using quantum mechanisms to function, it probably knows damn well how to deal with noise, with the cohering noise, right? So can we learn with nature how to deal with noise? I'm not saying, well, let's replace Google's beautiful computer with molecules, but is there some strategy, some like interaction with the environment that is actually being beneficial for nature that we can apply on top of those existing very nice quantum computing architectures? Or again, or can we start manipulating those spins in biology in a very predictable way so that we can alter, disease progression? We can alter those chemically spin-dependent chemical reactions to affect something. And we're not the only people thinking about this. There are many groups around the world that are taking this very high-tech approach towards quantum biology. But we think it's super exciting, and I'm not endorsing nature, but I'm endorsing this cover of nature, because I really think that interdisciplinarity is going to get us very, very far in the future. And to the left there, there's Doc Quantum, tiny Doc Quantum. Next time we play, I want to be Doc Quantum. So I want to thank my wonderful group members who decided to join our starting group under a pandemic in a field that is still controversial to some extent. So they're the reason I wake up every day. So this is what I wanted to tell you. I have a part about my professional development if you guys are curious, but for right now, I want to say that may the quantum be with you.
Thanks, Clarice. Thank you.
How do you want us to do? Shall we open for questions or shall we go to the professional development thingy?
So wait, I'm gonna stop recording.